Today I will be discussing glycolysis. Glycolysis is an important metabolic pathway that converts glucose into ATP. ATP is one of the main energy sources of the human body. Glucose is first phosphorylated in an irreversible step to glucose 6-phosphate. The red arrows will indicate irreversible steps, and there are a total of 3 irreversible steps in glycolysis. Glucose is converted to G6P via glucokinase or hexokinase. Glucokinase is found in the liver and the beta cells of the pancreas. Hexokinase is found in other tissues like the brain. The difference between the two that you should note is that glucokinase has a high Km and high Vmax, so it has less affinity for blood glucose and is in a way a glucose sensor because it senses elevated levels of blood glucose. Hexokinase has a low Km and low Vmax, therefore it has higher affinity for glucose, which makes sense because you find hexokinase in tissues like the brain where you need to have glucose as the main energy source. So it's important that this enzyme has a high affinity for glucose. There are certain inherited mutations of glucokinase like MODY2, MODY2, which is a rare form of inherited diabetes mellitus, where the beta cells of the pancreas don't exactly respond to high blood glucose levels in the way that they should. In any case, this step requires the use of ATP. So we're using 1 ATP to convert glucose to glucose 6-phosphate. Glucose 6-phosphate can go into glycolysis, and it can also go into other pathways as well, like the pentose phosphate pathway, also known as the HMP shunt. It can also go into glycogenesis and also be formed from glycogenolysis and gluconeogenesis. Glycolysis occurs in liver and tissues that don't have mitochondria or don't have oxygen, and also in the brain in both the fasting and the fed state, and it makes ATP in the presence and absence of oxygen. As you may have heard, there's aerobic respiration and anaerobic respiration, so glycolysis can be involved in both of those, and also in the presence or absence of mitochondria, like I stated. And glycolytic enzymes are found in the cytosol, so that's important to note because glycolysis occurs in the cytosol, but the TCA cycle, which I'll talk about in a different video, occurs in the mitochondria. So after you form G6P, G6P then becomes fructose 6-phosphate via phosphoglucose isomerase, and this is a reversible reaction, as you can see from the double arrows here. After that, fructose 6-phosphate becomes fructose 1,6-bisphosphate, a 6-carbon structure via phosphofructokinase 1 and the use of 1 ATP in an irreversible reaction. And PFK1 is an important enzyme and is the most important regulated step. It is an allosterically regulated enzyme, in fact. And let's talk a little bit more about this part of the pathway. So if you look over here, when there's high levels of blood glucose, there's high insulin, decreased cyclic AMP, and decreased pKa. Now there's a bifunctional enzyme, which is just one protein that has PFK2 and fructose bisphosphatase 2. When there's high insulin and high blood glucose, phosphofructokinase 2, not 1, gets dephosphorylated and gets active. Yes, active. And the other part of this bifunctional enzyme, the fructose bisphosphatase 2, becomes inactive. That leads to increased formation of fructose 2,6-bisphosphate in the heart, the liver, and the skeletal muscle. Why is this important? Why are we going through all of this to make fructose 2,6-bisphosphate? Well, fructose 2,6-bisphosphate and AMP allosterically activate PFK1 to allow glycolysis to continue. In the presence of glucagon, or low blood glucose level, this bifunctional enzyme is phosphorylated, so fructose 1-bisphosphatase will phosphorylate PFK2, making it inactive, and will activate fructose bisphosphatase 2. So this is kind of the opposite of what happened here. And fructose bisphosphatase 2 will degrade fructose 2,6-bisphosphate, and that leads to allosteric activation of fructose 1,6-bisphosphatase in gluconeogenesis. Over here I've written a list of factors that inhibit PFK1, including ATP, which makes sense because if you have a lot of ATP in the body already, why would you go through glycolysis and make even more, right? So in a way, it's negatively feeding back on the PFK1. Citrate, glucagon, because if there's low blood glucose levels, then you wouldn't be undergoing glycolysis, you would lean more towards gluconeogenesis to make more glucose since there's not enough. Fructose 1,6-bisphosphatase, fructose bisphosphatase 2, like I stated earlier, decrease in blood glucose, decrease insulin, and decrease in fructose 2,6-bisphosphate. So remember these factors. Now I drew out the rest of the pathway and we'll discuss each step one by one as before. But before we continue, I just want to note that this part of the pathway that I just discussed is the energy investment phase, which makes sense because you're literally investing energy, which is ATP. You're putting a total of 1 + 1, 2 ATP in to ultimately make you more ATP at the end of this pathway, which we will go through. So this is the energy investment phase. Now let's move on to the next phase, the cleavage phase. So you're cleaving a 6-carbon sugar into 3-carbon intermediates. So fructose 1,6-bisphosphate becomes glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. There are 2 of these made because from now on there will be 2 reactions occurring to ultimately give you 2-pyruvate, which we'll see later. And this step occurs because of aldolase A and B enzymes. Aldolase A is found in the muscle and aldolase B is found in the liver. And aldolase B is also required for metabolism of fructose in a separate pathway. And dihydroxyacetone phosphate can convert into glyceraldehyde 3-phosphate via the enzyme triose phosphate isomerase in order for glycolysis to continue using glyceraldehyde 3-phosphate. In the next step, 2-NAD plus becomes 2-NADH plus 2H plus overall. And glyceraldehyde 3-phosphate becomes 1,3-bisphosphoglycerate. And again, 2 of these 1,3-bisphosphoglycerates are made via the enzyme glyceraldehyde 3-phosphate dehydrogenase. This step can be inhibited by arsenate. I think there are 3 main enzymes to note that are inhibited by arsenate. That includes glyceraldehyde 3-phosphate dehydrogenase, alpha-ketoglutarate dehydrogenase, and pyruvate dehydrogenase. So they're all dehydrogenases. And in terms of the pyruvate dehydrogenase, the arsenate specifically interferes with the lipoic acid. 1,3-bisphosphoglycerate then becomes 2 of these 3-phosphoglycerates via phosphoglycerate kinase. And here is the first step where ATP is formed. I only wrote 1 ATP because I wanted to show you that in one reaction, 1 ATP is formed, and in 2 reactions, there are 2 formed. So, like I said earlier, from the fructose 1,6-bisphosphate step, after that, you'll have 2 reactions. So in total, for this step, you'll have 2 ATP that's being formed. And this occurs via substrate level phosphorylation, which is important during periods of low oxygen, in other words, hypoxia. And you wouldn't need to use the electron transport chain in phosphate levels phosphorylations. And you can think of 1,3-bisphosphoglycerate as a high energy intermediate molecule as it forms this ATP. So I guess think of it as this molecule has so much energy that it even gave off an ATP. The 3-phosphoglycerate becomes 2-phosphoglycerate via phosphoglycerate mutase, and 2-phosphoglycerate becomes phosphoenol pyruvate via enolase. And the final step, phosphoenol pyruvate becomes pyruvate via pyruvate kinase in an irreversible step, also forming ATP and a total of 2 ATP, which I'll write in at the very end. So what did we form from this entire glycolysis process? We formed 2 pyruvate molecules, which will go into TCA cycle later on. I put stars next to pyruvate kinase and phosphoglycerate kinase to indicate that ATP is made in these 2 steps and that there's substrate level phosphorylations. So what is the net outcome of glycolysis? Well, we formed 2 pyruvate molecules. We formed 2 ATP here, 2 ATP here, so 2 plus 2 is 4 ATP. We used 1 + 1, 2 ATP in the energy investment phase. So basically we got 4 ATP minus 2 ATP, which makes 2 ATP. But let's not forget the 2 NADH plus 2H plus here. How much ATP does NADH plus H plus give you? Well, 1 NADH plus H plus gives you a total of 2.5 ATP. And if you want to round it, you could say that 1 NADH plus H plus makes 3 ATP to make the math a little simpler. But if you multiply the 3 ATP by 2, because there are 2 NADH plus 2 H pluses formed, then you get 6 ATP. So 6 ATP plus the 2 ATP that's formed from the 4 minus 2 gives you a total of 8 ATP formed in glycolysis. So you get 8 ATP from one glucose molecule. And that's that. And I think the best way to tackle glycolysis is simply by drawing all of these steps out yourself, as well as the factors that influence the enzymes, like the factors that inhibit the enzymes and also activate them allosterically, for example. And then maybe the next day try drawing it yourself without looking, and then check to see where you made mistakes or if you forgot anything. That would help you to memorize this pathway and hopefully also understand it as well. But just make sure that you get glycolysis down, because there are other pathways that are also interconnected in many ways, including gluconeogenesis and the pentose phosphate pathway. Definitely get glycolysis down, because that will help you with seeing the overall picture and how everything is connected and related to one another.